Before there's too much entropy in the room, I'd like to keep on going with our imaging section. My name is Pete Choyke. I'm the director of the molecular imaging program at NCI, an MD. I'd like to thank the organizers for inviting me to speak, and thanks for including imaging in this workshop. As imagers, especially human imagers, we appreciate the large size of the animals we're talking about today, canines, and realize that so much engineering has gone into our ability to make very great high-resolution images at minimum doses on the human side. Typically, when we deal with mice imaging, we're dealing with customized imaging equipment that really is quite different, sometimes better in some respects, but also sometimes worse. The best example of the difference is CT, which is a low-dose, very rapid technique in humans. It's a high-dose, extremely cumbersome enterprise in animals, just because of the physics of the detectors. So it is interesting that dogs are very right-sized for imaging, and there are a lot of applications. However, the Achilles heel of imaging is its expense, and so it really has to be a great application to justify the use of imaging on a routine basis. So we're going to have two presentations in this session. I'm going to be talking about PET agents, and my colleague Mark Dewhurst from Duke will talk about MR spectroscopy. And I'd like to begin first by thanking my colleague Amy LeBlanc for her support in all this. She's been amazing, and she has a great team that works with us. You know, at the end of the day, I have to look my own dog in the eye, so it's very important that we do this very professionally. So PET imaging is an amazing invention in medicine. Over the last 15 years, it's become a routine test predominantly based on this agent, fluorodeoxyglucose, FDG, which interrogates the Warburg physiology of the shift to aerobic glycolysis that occurs in cancers. It's been amazingly effective. Here's a case of colon cancer that's easily detected on the PET scan, very nonspecific on the accompanying CT, but once you know where to look, you can see the thick wall of the cancer and how the agent takes up in that. But FDG has been a really great gateway drug in the sense that it has allowed deployment of PET CTs across the country. It spawned an industry of delivery of radiopharmaceuticals that used to cost thousands of dollars per dose, now costing less than $200 a dose. A whole distribution system throughout the United States. By the way, I would just urge you to drive carefully as you drive on especially urban interstate areas, because there are probably curies of activity in the car ahead of you. So just be careful out there. So of course the rationale for the use of companion animals in PET imaging really recapitulates many of the rationales for everything we've been hearing about in terms of potentially being able to accelerate the development of drugs, but also in the case of dogs, accelerating the development of imaging agents. And likewise, creating situations where we can make decisions about whether to go forward or not go forward on drugs and imaging agents. I think it enables one to conduct realistic experiments without necessarily going all the way to an IND. As has been said multiple times, toxicity is relatively easy to ascertain as opposed to mice, and even to some extent people, because you can be more intrusive with the animals. And the size, weight, the PK and PD are all better suited to the human situation than they are to the mouse situation. There's an increased feasibility to obtain validation tissue. One might argue that the IACUC is a little bit easier to deal with than the IRB when it comes to sticking needles. I think it's very important, and of course it can be used in the context of real clinical trials that we've been discussing today in pet patients. Just to do a slight comparison from the imaging perspective of animal models, companion animals versus mice models. Of course, I've said already that the canine is of more realistic size and fits in a human scanner. The geometry is ideal for that, and sort of equates to a pediatric patient for which these machines are very well designed. Whereas on the mouse size, we're dealing with a very small size, several centimeters, relatively large tumor, and devices like the micro PET CT that are very cumbersome, especially on the CT side, but even on the PET side as well. These are spontaneous heterogeneous tumors with stroma, whereas the cell lines that we do for mice are often only mixed with stroma at best and often prone to necrosis in their center, not very realistic from an imaging perspective. We do have an active immune system in a companion animal, and typically in most of the mouse models, we have an immune-compromised host. So the tumors are suitably heterogeneous in appearance, and we get to get a feel at the imaging level what kind of differences we'll see between highly active parts of the tumor and inactive parts of the tumor. Whereas in the mouse models, we either get a homogeneous lesion because there is no necrosis, or a kind of donut-looking lesion because of central necrosis that's really a black or white situation. The growth rates are more realistic versus the mouse models that can get really out of hand with regard to their growth rates. And the physiology is much more akin to the human, with heart rates on the order of 100 beats per minute as opposed to 300 or 400 beats per minute with rodents. So overall, there are reasons to be very enthusiastic about using companion animals in this setting. But there are a number of challenges of testing PET agents in this, with human equipment in these settings. So it's very important to understand the fundamental aspect of PET agents, a little bit different than drugs, that we rely on a high target to background ratio. So we want to get the agent to the target, but we also, very importantly, want to suppress it from the background to obtain this tumor to background ratio. The mouse models that we use tend to overestimate target affinity because we're targeting generally human tumors in a background of 0 target in the mouse background. And we also underestimate background because there's nothing for these targeted agents to bind to, whereas companion animal tumors are polyclonal and stromal. The background that we see is very much related to the pharmacokinetics, and the PK of rodents is very, very different than the higher order mammals. And so overall, the larger mammals provide us with a much more realistic view of the tumor to background ratio. So having said the virtues of PET, there are some challenges in the setting of scanning animals in an MRI, which after all, or MRI or PET, which after all is a tube, a gantry, where it's difficult to have monitoring. That is a penguin, by the way, that you're looking at from Amy's experience. So we have to be able to sedate the animals and have general anesthesia to facilitate the positioning and maintenance of the positioning throughout a fairly long time period of the imaging, which can go on for an hour or 2 hours if one wants to get a complete understanding of how the particular PET probe is functioning. And then there's the very important issue of radioactive waste during and after the scan. Animals are not toilet trained, and so we have a lot of problems to deal with in terms of maintaining a safe environment for our workers as well as for our patients who are going to be using the scans on a following day. So these are expensive pieces of equipment. They can't be just used for companion animals. They have to be used during the day for humans. Of course, in true veterinary research centers, that's not the case, but in a center such as ours, there's dual use. And as a result, we need to have a distinct separation of the schedule between the animals and the humans, and usually a 1 to 2 hour overlap or gap, so that there's no chance that a human patient will come in contact with a companion animal patient. And different people, of course, have different attitudes about that, but the assumption is that those shouldn't intersect at all. We need special staffing, not only the PET-CT technologist who will monitor the radiation during the procedure, but also trained veterinarian staff who is trained in the use of radioactivity and the dangers of using radioactivity, as well as imaging specialists. And that really moves to the issue of radiation safety. So radiation safety is actually our #1 issue in this environment. To use the technical term peeing and pooping, I hesitate to do that at the Institute of Medicine, but I really need to make that quite clear. And the concern is, of course, as you release the patient into the environment, they are going to do what they're going to do. And there are now more and more probes, sensors that detect radioactivity in the environment as a precaution against terrorism. For instance, the tunnels leading into Manhattan have sensors for radioactivity. And around this city and many other cities, there's monitoring for radioactivity, so that should there be a release of radioactivity outside of the normal environment, it could cause a lot of problems. Now, I should say that, what do we do with our human patients? Well, we often release them to their environment where they use the public sewage system, and it's not really an issue. It's below the regulatory limits that the NRC generally has any issues with, but if you're talking about the local street, fire hydrant, et cetera, there could be issues. So we do need to provide temporary housing after that. So, for instance, some of these agents, like C11, has a 20-minute half-life, clears essentially in 2 hours. It's not really a big deal. Gallium-68, 70-minute half-life. F18, which is our workhorse radioisotope, clears in 12 to 24 hours. But we have to really think twice about some of our longer-lived probes, like zirconium-89, which has a 3-day half-life, which would really clear in about 20 days. And you really have to really think about using that. And that, of course, would be matched with something like an antibody that would have a slow clearance over time. So that's some of our considerations from a chemistry point of view. Here's one of our patients in the scanner. So I want to talk about the types of agents that we might consider using in this setting. So broadly speaking, we can think about 2 different types of PET agents. Those that are very receptor specific, meaning either channel specific, or you get the meaning that it's highly targeted. And examples would be gallium-68 dotatate, which binds to the somatostatin receptor, or zirconium-89 panitumumab that binds to EGFR. In general, these are a little bit more difficult to work with in animals because, as we've heard multiple times, the receptors are not exactly the same. So the agent has to be reworked to, to accommodate the differences between the canine receptor and the human receptor. Now, that entails a whole different radiochemistry. Oftentimes, the radiochemistry for the human version will not work exactly for the canine version. So it's really a much bigger deal to work with specific agents. So we tend to use what I would call hallmark tracers, not after the card, but after Dr. Hanahan and Weinberg hallmarks, which are general final common pathway effects that you will see in cancers. The best example being FDG for Warburg, which is in common use, but others that might look at hypoxia, apoptosis, and proliferation. So I wanted to give some specifics in cases that Amy has encountered. So this is a 6-year-old mixed breed dog with B-cell non-Hodgkin's lymphoma and an FDG baseline showing a lot of adenopathy in the mediastinum and in the abdomen. And after conventional therapy, you can see that all of that resolves quite nicely on the FDG. And that's an example of a PET agent that's very useful for veterinary medicine. A more exotic agent is the agent FLT, or fluorothymidine, which was actually an analog of the drug AZT, but would prove to be a little too toxic for treatment of AIDS and HIV. But it is a marker of proliferative activity based on the nucleotide uptake. So the accumulation in cells is dependent on the activity of thymidine kinase 1 and the activation of the salvage pathway. So this can be an example of an agent that's very useful clinically. It's been useful clinically in humans, but it's also useful in the veterinary population so that it could be used in clinical trials. So in this situation where we'll use both tracers, the FDG and FLT, in a 9-year-old male castrate mixed breed dog, there was a mass identified on ultrasound and an FDG scan was performed. And you know, I'm beginning to wonder whether I'm violating HIPAA now with using the name, but there is uptake, a large amount of uptake, but it's actually fairly necrotic looking, and there's some, you know, issues, but there's also a focus of uptake in the mediastinum as well, and that would imply that perhaps this lesion is metastatic already on FDG. However, we found, and you can see this again in the sagittal view, here the CT and here the PET showing this area of activity in the mediastinum. But just like in humans, FLT has proven to be more specific for cancer. And when we look at that, on FLT, you see much greater activity in the proliferating parts of the tumor. And when we specifically look for that site in the mediastinum, it's not there. And this proved to be a hyperplastic lymph node in that area. So it can be used as a means of differentiating tumor from non-tumor. So there are a variety of ways that you can use these PET agents. Here's an example of an agent that I think may be very useful for human studies. So work in animals will be a benefit to humans, and that is this tetrafluoroborate, which is basically a marker of the sodium iodine symporter protein. So this is very active in the salivary glands, thyroid, and it's also seen, as you can see here, in the stomach, where the symporter is very active. But of course, we're very interested in the side effect of radiation on the salivary glands. It's an extremely debilitating symptom caused by radiation therapy. And in my own case, it's of interest because we can target radiotherapy using therapeutic radioisotopes, but at the expense of destroying the salivary glands. But we can study blocking the salivary glands, making them hibernate, blocking the symporter with an agent like this in dogs. So here's an example where the dogs can be of great benefit to the humans. I was interested by the last session where we talked about apoptosis, because there's a very good apoptosis imaging agent that could really benefit from experience in canines. As was stated before, there are two pathways of apoptosis, the extrinsic and the intrinsic, but they converge on caspase-3. And so an agent has been developed that's very specific for caspase-3 as a substrate as an enzyme substrate. So they looked at a variety of different enzymes here normalized, and only caspase-3 had significant activity. So this is a very rationally designed PET agent that has an elegant design. It has the 18F label on this active part where the enzyme substrate is contained, and then what they term a PEG, but in fact it's really something that makes the agent very lipophilic and therefore penetrates into the cell, whereupon this lipophilic component is cleaved by caspase-3. And this polar hydrophilic agent is retained in cells that are undergoing apoptosis. And this has been shown in various mouse models and correlates fairly well with markers of apoptosis. But because it's expensive to develop these agents, they've only gotten as far as normal non-human primates that don't contain tumors and haven't been treated, and even some human volunteers that were done in Europe. But again, while there have been no safety signals, there's rapid excretion. It's actually a very nice distribution of activity. Of course, the kidneys and bladder aren't going to be imageable, but other organs will be quite visible if it's true that it actually shows apoptosis in man. So this would be a very good agent to try in the setting of a canine tumor treated with an apoptosis promoting agent. So these are just some examples of where I think the field could be very useful. Both fields could be useful to each other. I think there are some open questions. Are animal patients recognized? Are the results recognized by the FDA? Will this really help us to approve these imaging agents? Or will the metabolic differences be considered by the agency rendering it not useful as an efficacy marker? So we're going to start with small but relatively commonly available PET agents and proceed to more exotic agents with time. So just to conclude, we feel companion animals offer many potential advantages in developing new PET agents, specifically because they're in larger animals that are heterogeneous, contain stroma. There are some unique aspects, challenges to the clinical environment that we're going to have to overcome. And for now, we're going to use the so-called hallmark tracers, final cascade tracers that are more likely to be relevant than the receptor or transporter specific tracers because of species differences. So we look forward to using PET agent testing in companion animals, and we hope that it'll be very useful in the future. So I'd like to thank you for your attention.